Our next speaker is Zahir Shah from the City of Hope, and you've heard of CAR T cells, and now you're going to hear about CAR macrophages.
Good afternoon, everyone. Thank you so much for inviting me here for giving this talk. So my topic of today is to generation of the iPSC CAR macrophage for cancer immunotherapy. And the idea of this came from the work done by Sargil in the University of Pennsylvania. They use the CAR macrophage derived from the PBMC and engineer with CAR, which reward their anti-inflammatory nature to pro-inflammatory, and they showed that engineering the macrophage with CAR can enhance its phagocytosis. And they showed that in vivo model that targeting the solid tumor, because being as a component of the solid tumor, the macrophage can easily penetrate into the solid tumor, can retain there. So they get this idea and engineer the macrophage and give them to the model and targeted the cancer cells. And we can see from the survival assay that it significantly enhanced the treated mice. However, this was from the PBMC isolated macrophages, and there were a lot of challenges. We cannot use it as off-the-shelf product. We cannot manipulate this at a single cell level. And another problem was they used a very high MOI for transduction for the adenovirus. That is 1,000 MOI compared to the 1 to 5 MOI we use usually. So in order to overcome these challenges, we use iPSC-derived macrophages, and we took the HSPC cells, reprogrammed in our lab, and we characterized in vitro and in vivo. And after this, we wanted to see which CAR should be used to, for these macrophages. So our previous study have shown that pancreatic tumor cells express high PSCA CAR compared to the positive adjacent cells. We can see the weak expression of the PSA antigen as well. So, and it also showed the survival assay that high PSCA is associated with high mortality. So using this PSCA antigen, we constructed anti-PSCA CAR and hypothesized that targeting this PSCA antigen on the pancreatic tumor cells with the CAR embedded with the anti-PSCA. And we also engineered membrane-bound IL-15 in our CAR product in order to activate the nearby NK cells against the tumor. And as an antigen-presenting cell, the macrophage can phagocytose the tumor cells, and then it can also cross-present this antigen to the naive T cell, and by this way, it can activate the naive T cell against the pancreatic tumor cells. So this was our approach, and we constructed the CAR with Mark, and our CAR, like I said, is anti-PSCA CAR, engineered with membrane IL-15. We transited in iPSCs and sorted the cells, and then we also selected the tumor cell, pancreatic cell line, which expressed high versus low PSCA antigen. Like we can see from here, this is the PANC1 we use as a PSCA low expressing cell line, and MIAPACA2 and CAP11, which express high PSCA, we use this cell line in our study. When we differentiated our cells into macrophage and co-culture this cell line, after labeling with CFSE, we can see that after 3 to 6 hours, we can see with the mark compared to the CAR. So the CAR has very high phagocytosis compared to the mark in the Kaplan-1 cell, which express high PSCA. And similarly, the same result was reflected in MIA-PaCa2, which expressed high PSCA. But if we came to the PANC1 cell, which expressed low PSCA, the mark in the CAR cells were almost the same as phagocytosis. There was no significant difference we can see from here, which shows that our CAR is very specific and which we can use in vivo for targeting the PSCA positive pancreatic tumor cells. So like what I said, we also engineer with membrane IL-15. So we showed in our study that incubating the NK cell with the CAR-iMac cells, we can see here, can significantly upregulate the expression of CD69, an activation marker on NK cell, compared to the untreated iMac cell, untransduced iMac cell, and compared to the NK cell only. And similarly, we also shown that co-culture CAR-iMac with Kaplan-1 cells, we can see here from here, over 5 days, we can see the upregulation of the CD69, CD8 positive memory T cells, naive T cell. And this was confirmed further by the expression of high expression of HLA molecule, because HLA molecule is actually presenting the peptide to the T cell. So this shows that after phagocytosing the whole cell, the tumor cell, it can take some time for the macrophage to present this antigen to the naive T cells, which we can see from this graph. Then we went to the in vivo model, and we get the strategy to inject the cells on different time points, like day 0, when there is no metastasis, day 3, early metastasis, and day 7, a little bit more mature metastasis, because the model, the Kaplan-1, MIA-PaCa2 was metastasizing. It can metastasize to the liver, the spleen, and the bone marrow, so we use these 3 time points. And we can see from the in vivo imaging that on day 3, before injecting the macrophage, so this was on day 0, we can already see on day 3 that it has worked. But it was before injecting the macrophage. We can see on day 3, day 7, we have the metastasis of the tumor. So after injecting on day 3, taking this image, we can see on day 7 that it also worked on day 3. But compared to day 0, day 3, the day 7 effect was less. And it was probably because the Kaplan-1 cell was multiplying over time, and we used the same amount of macrophage. So in order to get the effect of the macrophage, we hypothesized that we should increase double or triple the amount of the CAR macrophage to suppress the tumor metastasis. And this can be seen from the graph, the survival graph, that it enhanced significantly the survival of the treated mice compared to the day 7 and untreated mice. This was day 3 and this was day 0. Then we also checked the toxicity of our CAR macrophage compared to the T cell. Like T cell is known that it works very well, but it also causes the CRS, cytokine release syndrome, which the main challenge to the CAR T field, and it also caused the GVHD, graft versus host disease. So we injected the CAR macrophage, and after 2 days of each injection, we collected serum from the blood, and we sent to the company, and we see that in the treated mice, compared to the tumor only, we did not see any elevated level of IL-6, IL-1 beta, interferon gamma, and serum amyloid. These are the classical markers used for the CRS. Similarly, we also checked the GVHD in the humanized mice, and we can see here this was the mice treated with the CAR macrophage compared to the tumor only, and we did not see the accumulation of the lymphocyte, and we can see some accumulation here in the tumor level in the tumor only, but not in the CAR macrophage, which reflect that our data is consistent. And we didn't stop here. We also collected the abdominal fluid because we injected IP, intraperitoneal injection of the CUP1 cell. So we also collected the abdominal fluid and we sent for the cytokine, and we can see again we did not see accumulated high expression of the CRS-related cytokine, which shows that the macrophage, CAR macrophage, compared to the CAR T cell, it has minimum toxicity. We will not say zero because we don't know. It needs large cohort study to confirm it. Then there was another problem in the field before the people, they published the in vitro data only, and they showed that during differentiation, the CAR retention is a problem. When it goes from iPSCs to progenitor and then mature, so the methylation happens in the promoter region and the CAR expression is going down. So we tried to overcome this problem, and we single cell sorted. We isolated monoclonal cell, CAR-positive iPSCs, and then we differentiated this. And you can appreciate that at day 0, we can see all the cells, they are positive, and on day 10, we can see that almost 99 to 100% cells, they retain the CAR expression. But differentiating the progenitor cell into macrophage, it decreased the level of CAR expression almost 20%, and we have expression of the CAR at around 80%, which is still very high compared to the published protocol. And we can see from this graph easily that iPSCs retain the CAR expression and the progenitor cell retain, and when it's going to differentiation, so the CAR level goes down, but in our monoclonal iPSC cell, it did not go down below 50, like what people said 30 and 20, so we seen 80% CAR expression at this level, and which was also the success in our story. Then there was another issue that we cannot cryopreserve the macrophage itself because it's fragile, and if you just freeze them down and then you take them out, so you will see almost all the cells, 80 to 90% are apoptotic. So how to overcome this problem? We also tried the macrophage as well, but then we got the same result, and we tried the progenitor instead of the mature macrophage. So at progenitor level, which has the expansion power, which you can also expand after a thaw, so we checked the fresh versus 2 months cryopreserved macrophage, and we saw that these cells, these progenitor can retain the CAR expression, plus these cells can be differentiated into macrophage. So it can make these cells as an off-the-shelf readily available for the treatment. So after macrophages are differentiated from the cryopreserver, we also did the functionality assay, and we can see compared to the bulk cells versus the monoclonal cells. So we can see a clear difference in the phagocytosis, which is very high in the monoclonal cell compared to the bulk. And then we can see with the mock. And this result can be seen from this graph as well. So with this, we published this work in the Cell Stem Cell, and we concluded that human anti-PSCA macrophage can be used against the pancreatic cancer for the treatment, and we are working on this to further enhance this, this product for the clinical. So with this, I will acknowledge my lab members and mentors. So this is our mentor, Dr. Caligari, Dr. Yu, who supervised this study and we carried out. And many thanks to the CIRM who support me for the fellowship, and also the USC for arranging such a wonderful conference. Thank you so much.
Quick question. So you're saying there's cross-reactivity between the macrophages and the T-cell populations. Why wouldn't there be higher levels of interferon, gamma, and beta signaling if there's that cross-reactivity?
So what I understand your question is you're saying when it activates the naive T cells, why don't we see the CRS, right? Related cytokine upregulation. So these are naive T cells, so it's not a kind of expansion. The problem with the CRS is when you are injecting the CAR T cell, which are already CAR T, which has very high expression. So you cannot compare the artificial CAR because CAR is very high compared to the antigen cross-presented cell, which is very low, the level. This is one answer. Second, when you inject a bulk amount of the CAR T cell, let's say 2 million, 4 million. So within 2 or 3 days, within 1 day, it expanded very quickly. When it found it attached to the antigen, so expanded very quickly. But the naive T cell are expanded slowly. They are activating slowly by the CAR macrophage. So they are not going to expand that quickly like what the artificial, the CAR expression is very high, so it expands very quickly. Thank you. Thank you, sir.